I see your slides.
Okay, good. You can hear me just fine?
Yep, I can. Everyone?
Okay.
Good.
Excellent. Then I'll begin. All right. Well, hello, everyone, and thank you for sticking around for the last talk of this wonderful workshop. I just want to thank Hollis, Michael Orr, and Silas for inviting me to present. I think this has been an excellent workshop, and I look forward to sort of sharing some of my thoughts here on the future of bee taxonomy. My goal is sort of like to paint a picture of sort of, I think, the trajectory of things, and some methods and tools that I think, currently are important and will sort of increase in importance over time. And, the sort of title is supposed to be just the future of bee taxonomy, but I added, and identification to the title because I sort of realized, and I've sort of seen in this discussion, in this workshop so far, that the word taxonomy can mean different things. And so for many, I think taxonomy means just identification. However, I think one of my arguments in this talk, and I think sort of a need we need to think about is that taxonomy to taxonomists is much more comprehensive. And that only by doing better taxonomy, I think, can we produce better identification tools. And I'll go into a bit more detail about that. But I was also asked to sort of, by the committee to kind of paint an upbeat, and positive picture of the future of bee taxonomy. And thankfully, I think as a practicing taxonomist, just in terms of sort of, the tools and the resources we have available to improve taxonomy, I'm generally very optimistic and excited about taxonomy as a science in the modern era. Over the last decade or two, taxonomy has undoubtedly been sort of reinvigorated by these, many technological advancements. And so these include things like advancements in DNA sequencing, specimen imaging and data digitization, databasing, computing and bioinformatics tools, and I think also very much social networking and collecting, sort of improvements in collecting and people getting out. And I think this workshop is an example of this sort of, latter sort of networking and communication. I mean, through Zoom, we're able to connect with, have or conduct valuable meetings with bee taxonomists from across the world. And we can also use social media, I think, like iNaturalist, to make discoveries and tap into citizen science contributions that in the past we couldn't do. And so all these things sort of help us, I think, speed up, the science of taxonomy, as well as I think do more impactful science and research. I think it's also particularly exciting to be a bee taxonomist in the current day. So this is, you kind of mentioned in another talk with real data. I'm sort of presenting a cartoon, but it's based on sort of a recent entomology or talk at the entomology annual meeting in Vancouver, where Jamie Strange and Margarita Lopez Uribe presented data showing that over the last 20 years or so, interest in bees, at the ESA meetings and in departments across the, the US, has sort of interest in bees and other pollinators has increased exponentially, sort of probably matching, concerns over insect decline and pollinator health generally. I think because of this interest, bee taxonomy and taxonomists have become an increasingly important commodity, and the perceived value of bee taxonomy is increasing. Thus, in my view, now is a great time to be a bee taxonomist. And the challenge, though, is to capitalize on this moment before it's too late and the interest, goes away. So again, what is bee taxonomy? A question that others have posed. Although this was discussed in previous talks, I want to restate the goals of taxonomy and how the word taxonomy can have different meanings. So for the practicing taxonomist, the word describes, I think the objectives and goals of discovering, describing, and classifying species diversity. I think the field also, includes additional aims like, distribution mapping, natural history observation, and also increasingly phylogeny inference. And I think one of the goals of taxonomy is sort of species identification, but that comes as sort of naturally as a part of the process of delimiting and diagnosing species. I think for non-taxonomists, which we've used that term multiple times now in this workshop, so including people in this monitoring group, I think taxonomy is sometimes restricted to really just focus on this topic of identifying species. Thus, I think taxonomists and non-taxonomists have slightly differing needs, with taxonomists wanting data and tools to better delimit species and conduct revisions, while non-taxonomists really solely want, tools and resources to accurately identify species, usually in order to answer some questions. So I think consequently, taxonomists, are the ones who generally create the baseline resources, needed for species identification, and the non-taxonomists are generally the ones sort of trying to use those tools. And I think that more and better taxonomy ultimately will result in better identification tools that will help everybody. And I think, something that hasn't really been shown is sort of the overall kind of workflow for taxonomy in general. And I think it's sort of valuable for people that aren't doing revisionary work to really look at and think about to some degree. And so this slide compares the workflow between taxonomists and non-taxonomists who need species level data. The taxonomic process, I think, I'm not going to go into detail of all these points, but I think the taxonomic process is increasingly integrative and iterative, combining data from multiple sources, including DNA, morphology, behavior, and distribution, for the purpose of delimiting and describing species. And this flow chart, on the left sort of, shows some of the key steps in the taxonomic process. And I think a really important one that we haven't really discussed much in here is sort of the, the examination of type specimens. And then of course also, the identification, the creation of identification resources, and that can vary a lot, from product to product. Whereas sort of the non-taxonomist or ecological workflow, is more about, designing a study, acquiring specimens, identifying them, and answering questions, but less on really the, producing, taxonomic products. So how has bee taxonomy evolved over time? So taxonomy has become increasingly more integrative, combining more types of data to help delimit species. I think most importantly, the integration of molecular data into taxonomic decisions has become commonplace. So taxonomy has become also more phylogenetically informed, with revisions being clade-based rather than, sort of geographically based. Tools for taking images and creating keys have improved dramatically. And the products of taxonomy have become more accessible, I think thanks to, PDFs and online resources such as Discover Life. And lastly, I think access to specimens has increased, probably exponentially thanks to connecting and international networking and tools like this workshop. And so I've identified, in thinking about this, sort of 6 key components that I think are likely to impact and hopefully improve the future of bee taxonomy, in the present and into the future. So these are, the integration of phylogenomic data, generation of sort of fast and cheap DNA barcoding data, use of environmental DNA and metabarcoding methods, increased digitization, combined with, artificial intelligence approaches, automated sample processing machines, and also the further development, I think importantly, further development of online taxonomy and identification resources. So I'm going to go now over each of these in a little bit more detail. And I'll just say that sort of my use of the word taxonomy, I think, kind of goes back and forth between kind of doing taxonomy versus, identifying specimens. And so some of these things apply more to one than the other. So I think the integration of molecular data and systematics, really has revolutionized the field over the last decade and continues to do so, and probably is really one of the most important and will continue to be one of the most important kind of aspects of, taxonomy into the future. And this is all driven by the fact that, sequencing costs, really have plummeted, over the last 20 years, making it increasingly easy to sample genes and even entire genomes, from specimens at increasingly lower costs. And so DNA sequence data, I think have and the importance of these molecular data is they really have multiple applications, that include sort of use in taxonomy. And so you can use molecular data for things like in aiding species delimitation, identifying species, and also inferring phylogenies, as well as other things like creating, looking at genomes and population genetics. And the methods have advanced greatly, over, since I've been involved in taxonomy, starting with sort of single gene, sequencing, to multi-gene sequencing, transcriptomics, and now doing full genome sequencing. And the choice of method really depends, on trade-offs between cost and throughput. So how many specimens you want to do, with cost quickly becoming, less and less of a consideration due to the advancements in sequencing technology. And I'll argue that, right now, the current gold standard, is the sequencing of sort of genome scale data using multiplexed next generation sequencing approaches. One of the most popular, there's various versions of this, and one of the most popular approaches is the targeted sequencing of ultra-conserved elements, a method that allows for the affordable sequencing of thousands of loci at relatively low cost, about $30 per specimen. The approach provides phylogenetic resolution at multiple evolutionary depths and has enabled the routine sequencing of museum specimens that are over 50 to 100 years old. And I think these museomic methods, approaches really enhance the value of specimens and research collections and potentially allow for the sequencing of critical type specimens that inform taxonomic decisions, and help us create, names and apply those appropriately to specimens. And so the approach also allows for the capture of traditional markers like the DNA barcode gene, cytochrome oxidase 1, which I'll talk about more in a minute. And so here's a figure from a recent paper I published in which I used UCE data, 2000 loci, to resolve the phylogeny of the subgenus Osmia, and also to examine species boundaries using extracted CO1 data from the UC data combined with data on the public repository BOLD, the barcode of life data system. And so second, I think sort of complementing phylogenomic methods is, the affordable sequencing of the DNA barcode gene CO1. So although phylogenomic data are more powerful and have more sort of uses, 658 base pairs of the mitochondrial barcode gene CO1 can often be enough to sort species and associate sexes. The tool can also be used to identify species and enable environmental DNA sampling. So DNA barcoding definitely has limitations. It can fail to delimit species in some cases. And it does not reliably recover phylogenetic relationships above the species level. And so I highlight here a great paper by Jason Gibbs. It's really one of the few articles we have looking at the overall value of barcoding in a group of bees, in this case, Lasioglossum, and really finding that in some cases it doesn't delimit species well, but in other cases it does. The method also fails to identify species accurately when there's not a curated reference database of sequences. So I think a really key part of this is that these barcode data can help you potentially find or to discover species, but for identification purposes, just like a reference museum collection, you need to have a reference database of properly identified and curated sequences. And so the trade-off really is that sequencing CO1, so the reason, even though despite these limitations, the trade-off is that sequencing CO1 in recently collected specimens can be done cheap and fast, and new methods that use the handheld Oxford Nanopore MinION sequencer have been shown to generate barcodes at a cost of about 10 cents per specimen, enabling super cheap and fast data generation. So you can sample a lot of specimens quickly and for not very much money. And another advantage is that there is a large online barcode database that exists, and thanks to the efforts of people like Lawrence, has grown and continues to grow rapidly for bees. But I do want to point out, that even for bees, a group that's been kind of a focal group for a while, only about 33.5% of known bee diversity in the US has been sequenced, at least according to sort of the public databases. So there's certainly some unpublished data that I'm sure that Lawrence probably has access to. But there's a lot of gaps to be filled in to really make it a complete reference library. So for molecular methods, to sort of sum up, I think in my view, the future of molecular bee taxonomy, sort of involves sort of a combination of approaches. The DNA barcoding of many individuals combined with sort of phylogenomic sequencing of a subset of individuals, sort of taking advantage of both approaches and certainly integrating morphology and sort of expertly ID'd specimens on top of that. So related to DNA barcoding, and this is sort of switching more to sort of just identification, are these eDNA and metabarcoding methods. So these two approaches focus on the rapid identification of species from environmental or bulk samples using metabarcoding approaches. And so there's different versions of this. So in one approach, bee DNA can be extracted directly, say, from the environment, like on flowers. Whereas in an alternative approach, bulk samples of bees, say from pan traps, can be ground up or even extracted non-destructively and then sequenced simultaneously. Both approaches have been shown to work, at some level. So we do know there is bee DNA on flowers and pollinator DNA that can be sequenced. And we also know that you can do these ground up samples and you can sort of sequence and separate, the CO1 from all these individual different, specimens and species and identify them to some degree. But a major challenge that I think a lot of people overlook is that complete reference data are lacking for bees. And we really need more validation, of these approaches with traditional methods. So there's definitely lots of examples now where people get DNA and do some degree of identification, but most examples, tend to be in sort of, less complicated, environments and more controlled settings, and often without the sort of validation in comparison to sort of those traditional methods. So that needs to be done before we can reliably sort of use that approach for bee identification. So expanding away from molecular approaches, I think the future bee taxonomy will be highly influenced by, continue to be highly influenced by specimen digitization and maybe more novelly, artificial intelligence approaches. So the creation of high quality montage images of bee species, including, more recently 3D models and CT scans, I think has greatly improved, sort of taxonomic revisions and species keys by making it easier to illustrate, diagnostic characters and also just the general gestalt of species. Also, by making all these images, more available online on websites, it's easier for people who don't have access to a collection to at least, to view a species that maybe they're trying to identify. Now, more at the cutting edge of just generating these images, I think has been, it has been shown that specimens can be potentially automatically identified from images using machine learning methods. So an example of this is the Bee Machine tool created by Brian Spiesman. This is similar to iNaturalist, but customized, in this case for bumblebees. So by training an artificial intelligence model using identified images of US bumblebees, Brian was able to create a machine learning model that was able to identify unknown images of bumblebees with about 92% accuracy, which is pretty good. And so the big challenge, however, is to extend this approach to other bee genera and regions, and also to see if we can make use of the approach on not only images of field specimens and large bumblebees, but of museum specimens, and much smaller species in general, like Lasioglossum. So the approach, although showing a lot of promise, is relatively untested and definitely need of development and sort of testing in other groups of bees. And the challenge is sort of, creating these image libraries, from expertly identified specimens. And so you really need, 50 to 100, images to train these models effectively. So it's a lot of effort and time to sort of create these approaches. But I think it definitely has a lot of promise. And I think an important research question moving forward is sort of looking at and comparing how this approach maybe compares to some of the DNA-based approaches, perhaps separately or combined together. So one of the impediments to bee taxonomy and identification, I think, is sort of sample processing, something we haven't talked about too much. But sort of with large surveys, often, in large surveys where there are lots of specimens collected, it can often take thousands of hours of time to get samples sorted and mounted, which sort of slows things down, even just, separate from the identification process. And so one solution could be something like this. This is a recently developed device called the diversity scanner. It was published in a paper by Whirl and colleagues. And this is the Rudolf Meyer group in Germany. And so the robot uses artificial intelligence and cameras to sort specimens by type. It then images the specimens and loads them into PCR plates for DNA barcoding. And then the DNA barcoding is automated. And using those DNA barcode sequences and images, you essentially can sort all the specimens into species bins or groups, for taxonomists then to look at and review. So far, this approach has only been developed for small insects and malaise trap samples and really focusing on flies. But there is great potential for this, for sorting pan traps and other commonly used bee traps. Lastly, I think the future of bee taxonomy will continue to involve the development and improvement of online taxonomic resources. So I think tools like Discover Life, that currently exist for bees, but I do think they're in need of improvement and perhaps more sort of user input to contribute to them. And so in my mind, the ideal tool would be something that integrates a biodiversity data system, it would integrate a biodiversity data system that focuses on bees and includes sort of varied resources and tools that would aid bee taxonomy identification. So one key thing that would greatly help bee taxonomy, I think, is the digitization and organization of all bee holotype images. And so I list a bunch of other sort of potential requirements for a sort of centralized bee website. So to finish, I just want to say again, I think it's a really exciting time to be a bee taxonomist, and a taxonomist in general, given the diversity of tools at our disposal and the interest in bees. And so I think what we need now is more taxonomy. The completion of a DNA barcode reference library, what we need is more integrative taxonomy. We need the completion of a DNA barcode reference library. I think we need digitization of more bee species, especially type specimens for the taxonomic process. I think we need development and testing of these AI-based tools for identification. I think we need improvements and expansions of the available web resources and online keys. I also think we need more training of sort of integrative bee taxonomists, as well as greater collaboration. My sense is that there's so much sort of, talent and resources and money out there, but sometimes the challenge is just saying, here is the problem, let's all work on this together to sort of solve that problem and create something that really benefits the entire community. And there are tools out there for other groups, like ants, where I feel like there is actually less interest and less focus. But for whatever reason, some groups have coordinated and developed things that the bee community has not. And I think now is the time to sort of, be a leader and do just that. So we may not have the bee tricorder just yet, but I think, we're getting closer, and there are a lot of new tools that are promising. And so with that, I will take any questions. Thank you.